Malignant tumor, what do? They are very fast in growth and they invade the neighboring cells also. And also there are a lot of volatile compounds which are used in various products that we use every day for cleaning and all that can also cause cancer. Of uncontrolled dividing cells that are found, it will be easily picked up and therefore cancer can be detected. Starts right from the meiosis, then it enters into the mitosis and the daughter cells are formed. Hello everyone, a warm welcome to the session for Class 12 Biology CBSE. I'm Dr. Divya, Biology faculty. So in this session, that is session 7 of chapter 7, Human Health and Diseases, we shall learn about a disease which is called as cancer, and we shall learn the detection and diagnostic techniques for cancer. So cancer is one of the most dreaded disease of human beings, and it is a major cause of death all over the world, right? So because of food habits, the mutations that happens in the genes due to exposure to various carcinogens that are present in the environment, nowadays a lot of people are prone to this dreadful disease and it's killing millions of people worldwide. So you all have learnt about cell division, right, in your first PUC or class 11th chapter. So in that you know that the cell division occurs in a controlled manner. That is, it starts right from the meiosis, then it enters into the mitosis and the daughter cells are formed. So we all learnt about that, and it is in a controlled manner wherein there is a proper communication with one cell to another. So when one cell completes its division, it will intimate the neighboring cell through signals. It will intimate the neighboring cells to start its division process. But in the case of cancer, that doesn't happen, and because of that, uncontrollable division and differentiation of the cells will start, leading to uncontrollable growth, resulting in the formation of tumors in a body. And in cancer cells, there is a breakdown of these regulatory mechanism, as I told you, and therefore, there will be uncontrolled growth resulting in the formation of tumor. So, cancer cells lose the property of contact inhibition. What is the word that we need to use there? Contact inhibition. So, what do we mean by contact inhibition? As I said, very soon as one cell completes its division process, it will intimate the neighbor cell which is in contact with it to start the division. But in the case of cancer cells, these cells will lose the contact inhibition process and therefore all the cells will together start uncontrollably dividing, leading to the formation of tumors. So therefore, cancers are nothing but formation of tumors in the body. And in cancer, that is especially the tumors, there are two types of tumor. One is the benign tumor and the other one is the malignant tumor. So what is benign tumor? So benign tumors normally remain confined to a particular location and they will not spread to other parts of the body, and therefore it is easy to remove them through surgery and by radiation therapy. So that is about benign tumors, and they cause very little damage to the body and it is quite curable. Whereas talking about malignant tumors are nothing but mass of proliferating cells, that is mass of uncontrollably dividing cells which are called as neoplastic or tumor cells, and these tumor cells, they grow rapidly through the bloodstream. They can go and invade other cells, spreading the tumors there also or creating uncontrollable division of those regions as well. So that is called as malignant tumors, and malignant tumors usually they grow rapidly and they damage the surrounding cells as well, or they invade the surrounding cells as well. So that is about benign tumors and malignant tumors. So these cancer cells, as and when they grow vigorously, they deprive the other cells of nutrition, therefore causing the damage to the other cells as well. So you all know about weeds, right? So in a garden, why do we remove the weeds? Because weeds are the plants which grow uncontrollably and they grow very fast and they spread or reproduce very fast to such a rate that they derive a lot of nutrition from the soil, therefore depriving nutrition to the plants, other garden plants which are present there. So that is why we remove the weeds, right? Same way here, cancer cells that differentiate and grow in such a faster rate that they take up all the nutrition, leaving no nutrition for the cells which are normal. So therefore, cancer cells, they tend to compete for nutrition or vital nutrients. And these cells, cancer cells, what they do is they can slow off or they can cut off from a particular region from where they are growing, and they can move through the bloodstream, and they can move to the bloodstream and they can get lodged or they can stick on to any other region in the body or inside the body, and they can cause cancer tumors there as well. So such spread of cancer we call it as metastasis, and metastasis is the property of a malignant tumor. Malignant tumor, what do? They are very fast in growth and they invade the neighboring cells also. They invade the neighboring cells means they take up all, because they are very fast in growth, they take up all the nutrients, depriving nutrients of the neighboring cells. And apart from that, they can just cut off or they can just detach itself from the region from where the tumor is present, and they can move to a new region and attach itself there and cause cancer, therefore spreading cancer into other parts of the body. And that spread of cancer from one part to other parts of regions of the body, we call it as metastasis. And metastasis is one of the property of malignant tumors. So that is why malignant tumors are hard to cure and it takes time to cure them. Next, talking about the causes of cancer. So cause of cancer, exact cause of cancer, till now it is not known, because people who follow a healthy lifestyle and who are not exposed to a lot of carcinogens and only depend on organic products also tend to get cancer. So cancer, there are some of the carcinogens are said to cause cancers. What are those? We shall look into. So transformation of normal cells into cancerous neoplastic cells. So neoplastic means they are abnormally differentiating cells or mass of cells that are there. We call it as neoplastic cells. So therefore, transformation of normal cells into cancerous neoplastic cells can happen because of physical, chemical or biological agents that we deal with in our everyday or day-to-day life. And these agents which cause cancer, we call it as carcinogens. So there are different types of carcinogens. So now talking about the physical carcinogens. So ionizing radiations like X-rays, gamma rays and non-ionizing radiations like the UV rays, they can cause damage to the DNA, leading to DNA mutation and thereby causing cancer. So the normal cells, they transform into neoplastic cells because of physical carcinogens as those that are mentioned. And there are some chemical carcinogens which cause cancer, such as especially tobacco smokes are known to cause lung cancer, and also there are a lot of volatile compounds which are used in various products that we use every day for cleaning and all that can also cause cancer. Next, talking about some of the viruses. Some of the viruses are also known to cause cancer. So one of the best virus which we can take as an example is the human papilloma virus, which is HPV, right? But nowadays vaccinations are available for girls who can take them. That is the HPV vaccine that is there, which can be taken to prevent cancer from that particular virus. So these viruses, they are called as oncogenic viruses. So the viruses that cause cancer, we call them as oncogenic viruses, and the genes that are present or the genome that is present in there, we call it as the oncogenes. And there are several genes which are called as cellular oncogenes or proto-oncogenes, which have been identified in normal cells. These usually they'll remain silent, but because of an exposure to a carcinogen, they can get triggered, they can get switched off, and they can make the cells cancerous. So when these cellular oncogenes or proto-oncogenes, when they get activated under certain conditions, they could lead to oncogenic transformation of the cells as well. So there are various physical, chemical and biological barriers which cause cancer. So physical, we talked about different radiations. Chemical, we talked about the tobacco smokes, which we took it as an example. And biological agents, some of the viruses, especially the sexually transmitted viruses, they are capable of causing cancer if not treated properly. And also there are some cellular oncogenes or proto-oncogenes which are already present in a body, which when triggered or activated due to some external factors, it can cause cancer in a body. So these are the causes of cancer. Next, talking about the detection and diagnosis of cancer. So early detection of cancer is quite very essential so that disease can be treated successfully in many cases. So cancer detection, there are various techniques to detect cancer. Among them, one is called as biopsy. The other one is histopathological studies of the tissues and blood and also bone marrow samples can be taken to count the number of cells present in the body. So we can count the number of RBCs, we can count the number of WBCs, and we can count the number of other unusual cells or cancerous cells present in the body to test for the cancer. So this especially, these tests are used to, that is bone marrow tests are especially used for finding out leukemia, that is blood cancer. So talking about each of these one by one. So biopsy. So what is biopsy? In biopsy, what they do is any tissue which they suspect it may be, or any tumor that is present in the body which they may think that it is a cancer, what they do is they take out or they cut the tissue into thin sections and they stain it and they observe the cells under the microscope, and it is usually done by a pathologist. So this is what is done in biopsy. In the case of histopathological studies of the tissue, they use techniques like radiography, wherein X-rays will be used, computed tomography such as CT scans will be used, magnetic resonance imaging, that is MRI, all these will be used to detect cancers of the internal organs. So these are the two techniques. Biopsy can be used to detect leukemia also, and biopsies can be used to detect a lot of other cancers. So cancers of the internal organ, histopathological studies on the tissues can be conducted. Now talking about computed tomography. So like computed tomography, wherein that is CT scan. In CT scan, what they will do is they'll use X-rays to generate a three-dimensional image of the internal organs of the body to detect for any abnormal growth. And in the case of magnetic resonance imaging, that is MRI, strong magnetic field will be created and non-ionizing radiations will be used to accurately detect pathological and physiological changes in the living tissue. So any abnormal growth or any mass of uncontrolled dividing cells that are found, it will be easily picked up and therefore cancer can be detected. So there are various other techniques also apart from using radiations, apart from that a CT scan, apart from using MRI, apart from biopsy. There are various other techniques also to detect cancer, wherein antibodies against cancer specific antigens will be used to detect certain cancers. So antibodies which are specific to a particular cancer will be used or will in a patient to detect certain types of cancers. And there are various techniques of molecular biology which can be applied, or genetic studies which can be conducted in order to detect the genes in individuals with inherited susceptibility to certain cancers. Because I told you, protogenes or oncogenes which are present inside the human body, they can get activated. So we need to find out whether that particular oncogene or protogene is present inside the body of that individual or not. For that, molecular diagnostic tools can be used, which you have already learnt in molecular biology chapter. And after identification of such genes, it will be helpful to treat that particular individuals in order to prevent the occurrence of cancers. So such individuals will be advised to not get themselves exposed to particular carcinogens to which they are susceptible. For example, tobacco smoke, etc. they can avoid. So what they will do is, first they will go in for genetic studies of the individual, and if they get to know that in their body they have the genes, or in their genetic material if they have the genes or oncogenes or protogenes which is capable of getting activated any time when exposed to carcinogens, so such individuals will be advised not to get exposed to carcinogens, so that they can keep the protogenes, that is proto-oncogenes or the oncogenes, they can keep it under an inactive form, or they can keep it switched off. So these are the different detection and diagnostic tools. So next we'll learn about treatment of cancer. So there is surgery, radiation therapy and immunotherapy are the main treatments that are used to treat the cancer. So in radiation therapy, what they will do is they will irradiate the tumor cells with certain radiations, and cells will be destroyed, making sure that the neighboring healthy cells which are there, they are not destroyed. So that is what in radiotherapy. And also several Chemotherapy is also followed, wherein in chemotherapy, several chemotherapeutic drugs are used in order to kill the cancerous cells. And some of these chemotherapeutic drugs that are there, they are specific to certain cancers only. And depending on what type of cancer is there, a particular chemotherapeutic drug will be used or will be administered through, that is intravenous administration will be done in order to kill the cancer cells. And these chemotherapeutic drugs, they can have a majority of side effects because a person who is treated with these chemotherapeutic drugs, those chemicals can also attack the immune, or it can also destroy the immune cells, therefore making the immune cells count less in the body of the cancer patient. So therefore, it can lead to a lot of, apart from that, it can also cause a lot of side effects like hair loss, anemia, that is lower count of RBC, etc. can also happen. There is one more treatment for cancer which is called as alpha interferon. So most of the cancers are treated by a combination of surgery, that is removing the cancerous part, by radiation, that is treating the cells or irradiating the cells with certain radiations to destroy that particular cancer cells, and one is administering chemotherapeutic drugs in order to destroy the cancer cells. But in some cases, what happens is certain tumor cells which are there or cancer cells which are there, they are shown to avoid detection and destruction by the immune system. That is, the immune system cannot detect and identify them. So that is the reason why our immune cells cannot act on the cancerous cells, because our immune cells is capable of only detecting those cells which are foreign, which has come from outside into a body, right? But cancer cells are our own cells which are uncontrollably dividing. So our immune system will think that, okay, these are the cells which belong to that particular person's body itself, and it will not attack it. So therefore, patients are given biological response modifiers such as alpha interferons, which can activate their immune system and help in destroying the tumor cells. So what they do, they give or inject the patient, that is cancer patient, they inject them with alpha interferons, which are nothing but antibodies, and these antibodies are capable of detecting the cancer cells in specific and destroying those tumor or cancer cells. So alpha interferon is also one of the treatment for cancer apart from surgery, radiation therapy and chemotherapy. So this was about the treatment for cancer. So I hope you understood the session well. So we got to know what are the two types of tumors in cancer and what is metastasis, and also we got to know what are the tools used for detecting and diagnosing cancer, and also we got to know what are the treatments used for cancer. So in the coming session, we will talk about drugs and alcohol abuse, what are the different drugs and how the adolescents are prone to these drugs and alcohols, and what can be done to avoid drug and alcohol abuse. So see you in the coming session with this topic. Thank you.